Right, good morning, good afternoon, and good evening, wherever you are on the globe. Welcome to our webinar focused on biopharma. And I want to thank you all for joining us today. And before I hand you over to the panel, I just want to point you out to the Q&A tab on the side of your screen on the right-hand side. Please do ask or type in any question that you would like the panel to answer. We have a dedicated space at the end of this presentation for questions and answers. And any questions that are not answered today, we will get questions and answers put together in a document, and in due course we will email it out to all the participants on the webinar today. So yes, thank you for joining. I'm going to hand you over to John and the panel of experts.
Thanks so much, Joe, and let me share my good morning, good afternoon, and good evening to everyone who's joining us today. We sure do appreciate the fact that you've taken some time out of your day to join us and attend this webinar. Obviously, with everything that's happening all over the world and the global response to COVID and trying to adjust to a new normal and the impact that it's had on our workflows and how we do our work and where we do our work from, we know that everybody has just a million things that they could be doing right now, and we truly are honored that you have made this webinar a priority, that this is something that you felt was important for you to dedicate a portion of your time. And ultimately, we want this time that we have together today to be something that you feel delivers value to you, where the tools that we discuss might very well be leveraged in your application and at your organization to help you gain some time back in your everyday workflow and also save some money in the process. That's really our goal. So that you know who we are Cole-Parmer, and we've been in the business of serving our customers and providing support and fluid handling tools for our customers for over 65 years. Our pumps and our tubing and those things coupled with quite a large array of other proprietary technologies and products. They're utilized all over the world, and really almost every single market that you can conceive of where fluid is being moved, whether it's in biopharma applications like what we're focusing on today, or in food and beverage manufacturing and processing, wastewater treatment, industrial applications, really any other number of other markets and those kinds of things. Really, our products can truly be found everywhere. And along with our products, one of the things that truly sets Cole-Parmer apart at a global scale is the in-depth product knowledge and the application support that our team of experts can bring to the table. We have experts that are stationed all over the world through our offices that are located in various countries, and all of us are together as a team ready to come alongside our customers to really truly help you achieve success in the applications that you have. I'm privileged today to have two of my colleagues with me, and we'll be co-presenting and sharing some of those responsibilities today. And just to help put a voice to the face, I thought I'd have an opportunity for them to share a little bit about who they are. So I'm going to start with Colin. Colin, could you introduce yourself for us, please?
So to reiterate John's, thank you for that, John. Good morning, afternoon, and evening to everyone listening. My name is Colin Heathfield. I've been doing pumps and fluid handling for approximately 30 years. I've worked on a global scale across many industries. Now, John touched on pharmaceutical. I've covered pharma, petrochem, biotech, food industry, and not just with one technology, looking at several different technologies. And that's my sort of approach as a humble mechanical engineer, to give the best solution to move the product you want to move. And I think that's probably why we have a very good support system that does help you as a customer. I've been at Cole-Parmer for 15 years, and I'm now the technical specialist for pumps and fluid handling for EMEA. And thank you for that, John. I think, Phil, your turn now. Are you there, Phil?
Yeah, Phil, if you could take yourself off mute, sir.
Can you hear me now?
We certainly can.
Goodness, I'm sorry. I apologize for that. Don't know what was going on. Yeah, thanks, Colin. My name's Philip Nyron. I've been with Cole-Parmer for 23 years now, and and I'm currently the global product manager for the MASTERFLEX and ISMATEC ranges of peristaltic pumps that Cole-Parmer manufactures. And thank you, everyone, for joining, and we'll get to the content now.
Thanks, Colin and Phil. Of course, my name's John Batts, and I've been involved in fluid handling in a variety of capacities for the last 29 years, ever since just around 1991 or so, while I was still at the university. And I've primarily been involved in pharmaceutical side of things, but have also been in aftermarket sales and management and category management at the commercial side of things, and providing training and focusing on really showing our customers how the products that we have can be leveraged as tools for their purposes in order to help them achieve success. And really collectively, just with the three of us, we have more than 80 years of experience. Part of that's because we're all a little old, and that's okay. You know, we might have some calendar years behind us, but we also have a lot of fluid handling experience and really passion about the products. That's what we bring to the table. We're very passionate about fluid handling and passionate about helping our customers move past whatever obstacles you might have in order to really achieve the success that you're looking for. So today, our focus is really on ways, again, that you can leverage some of the very innovative and unique technological tools that exist on the pumping world to help you make some process improvements at various stages of the biopharma workplace. That's our goal. And really to help you gain some time back into your calendar and save some money in the process. That truly is our biggest objective for the day, and to help you do that. And along with that, we'd really love to help you really maximize your production yield and efficiency by employing some tools in some of the single-use side of things. And also, we would be remiss if we didn't touch on some of those challenges and changes that we're all experiencing in our workflow and the working conditions and where we're working from as the whole world continues to adapt to fighting COVID-19 and really to help prevent its spread. Now, much of what was really driving our webinar today can be traced back to the rapidly changing landscape of pharmaceutical development. And for a long time, the big focus in pharmaceutical development has been on synthesizing drug compounds, utilizing a lot of very complex chemical processes and methodologies for production and testing. And it's been, this has been the big focus for pharma for quite some time. But over the last several years, some of that focus in pharmaceutical development has begun shifting to the biopharmaceutical processes and really all of the benefits that brings. Here, as most of you know who are joining us today, the companies that are involved in biopharmaceutical processing are companies that are leveraging either bacterial cells or mammalian cells and utilizing them as mini factories so that those cells can produce the target compounds of interest, either through replication or through replication and expression and doing those things that cells normally do. If we can tap into the genetic capabilities of those cells to produce the target compounds that we're interested in achieving, that's where the biopharmaceutical world really brings its power and allowing us to produce really complex molecules by leveraging cells to do our heavy lifting. So the shift in the biopharmaceutical world to this whole process really brings with it a lot of challenges and some things that we have to think through in order to really streamline how things function. And in these areas of the system of the way things work, those are areas where they can easily go awry, and they can take up too much time and cause the whole production process to be thrown off schedule. So with the standard workflow in mind, that's what we're going to be focusing on. I thought we'd take just a moment just to really establish a baseline by looking first at a very typical biopharma workflow and looking at it through the lens of the upstream process and the downstream stages that have to take place. So if you look at this image, it's very representative of what typically takes place in a biopharma workflow process on the production scale. And you can see that you go from cellular growth optimization and you move into the production process where you're trying to produce that target compound. And then at that point, once the compound has been produced, then you go through various stages of cleaning and purifying and filtration. and ultimately to packaging and storage of the product that's been produced to the process so that it can either be further tested or it can be it can go through the sales cycle and get out to the end destination. Now, generally speaking, it's an intuitive process to explain, but in practicality, there's quite a bit of complexity that's there. And in order to really understand those complexities and the challenges that can be faced, it's important to maybe look at the whole biopharma process in 3 different stages, and the 3 stages of the development cycle where production is taking place. So if you think about it from that perspective, we've got that first stage, which is the discovery and that early research and development stage where you're just determining how to produce what it is that you're wanting to produce. And then you get into the pilot plant stage where you're starting to scale up those production processes and get ready for true full-out production and overcoming some of those hiccups along the way that you might encounter, some of the things that might be introduced as you go to a larger scale from the benchtop scale. And then, of course, you've got the full mass production process stage where it's a completely different thing that you have to consider, lots of different challenges that can occur when you move into that full production process. So let's look at each one of these stages in detail. We're going to take just a moment at each stage along the way to consider a challenge that might be encountered and how you might be able to overcome some of those challenges through some tools that we have to offer. So in the earliest of production stages, that research and development stage, the goal is really optimization of the production recipe, that whatever it is that you're using to produce that new and novel compound that you've discovered. And obviously, in order to achieve that, you have to adjust that mixture of the media and make sure that you've got all the right components that are there, and adjust your environmental conditions that the cells are being exposed to, and simultaneously you have to monitor the health of the cells and make sure that the cells that are producing compound, that those cells are that they're producing the compounds of interest, but then also the cells that are not producing the compounds of interest, that their growth is being suppressed, so that you're really truly optimizing the whole mixture. That's really the goal that you're trying to achieve. And that takes us to one of the big challenges that we can encounter when we're in this stage. Challenges that are faced by scientists and technicians when they're trying to develop and optimize everything. And that is really, how do you optimize that the growth conditions, and do so by eliminating some of that potential for human error. It's a very labor-intensive process in order to tweak that media mixture and monitor all of the environmental conditions and test and check on the health of everything. It's just very labor-intensive. And because of all of those things that are happening, typically very simultaneously, there's a good chance that human error can occur simply because of everything that's happening. In order to help explain this, we can take a look at a hypothetical scenario. We've got Tanya Smith. She's a very typical R&D technician. And in any given day, she has to collect and process just scores of samples. And again, she's looking to quantify the growth rates and the effectivity of the growth enhancement process that's being employed, and the suppression techniques that are being employed. And of course, she has to monitor for contaminants all along the way. And between all of these setup and the tests that she has to run and the samples she has to grab and be able to process, she's generating a lot of waste, and she's also introducing the good potential for human error, just simply because of all the processes, those manual processes that have to happen. Some of the questions that she may be considering, and things that you may be considering as well as you're working through at this R&D stage, are, what is that best reagent mix that you need to achieve, and the feed rate for each of the ingredients of that mixture in order to maintain the right balance and to promote the growth that you're trying to achieve. And then also, how do you minimize that possibility of introducing contaminants into the mixture? That's always a big important thing to consider, so that you don't have those challenges. of a cell feed or a cell mixture that's being contaminated by some other competing product that's trying to grow at the same time. And then also there's that consideration of how could I possibly automate some of the steps that I have to take every day so that I can make some process improvements at this stage of development. And that's really ultimately my goal. And so in order to do that, one of the tools that we want to share with you is the Ismatec Reglo ICC. The Ismatec Reglo ICC is a tool that Tanya could consider and that you might be able to consider to help you automate some of those processes at the R&D stage and really to help you mitigate risk along the way. The Reglo ICC is a multi-channel low-flow peristaltic pump, and the ICC nomenclature that's there stands for independent channel control. And what that means is that the pump literally has independent, it's a multi-channel pump in a small compact size, but every individual channel can be separately controlled, both in rotation and speed and timing and function, and all of those things can take place separately. So it's like having up to 4 pumps in one. And in fact, we'll see a video clip here that that shares a little bit about, hopefully the video comes through okay, where you can see how each of the 4 channels can truly run independently. They're also separately calibratable, which is a nice feature for the pump because that allows you to make sure that each individual channel is delivering exactly the fluid flow rate that you're looking for or dispensing the fluid volume that you're trying to achieve. And again, because they're all independently controlled, then that gives you that capacity to do lots of very interesting things at an automation level. And in fact, because each channel is independently controlled, then you think about what Tanya could do and how this might impact what Tanya is doing in her job cycle. And one thing that Tanya could do is that she could selectively add those media ingredients that she's trying to use in order to achieve that optimal balance in the media mixture. So that allows her, if she's monitoring pH or she's monitoring how quickly a particular reagent is being consumed, then she can either add on demand that particular product, or she could do so, maybe it's something that's a suppression reagent that she can add in real time, very steady delivery, and not have to think about it. Once she's calculated out what that would need to be, she can allow the pump to do that work for her. And simultaneously, because the channels again are controlled independently, that means that Tanya could, while the pump is pushing fluid out to the mixture where the cells are beginning to grow, then that she can also draw samples from that same very bath in order to test to make sure that they're doing what they're supposed to do, to check for effectivity, monitor for foreign contamination, and even circulate back to the system if that's important. So there's a lot of opportunity for automation and process improvements along the way. So if you think through this at a practical level, if you think through the number of samples that Tanya might have the capacity to process manually on her own, maybe she only gets an opportunity to process 100 samples an hour during her daily shift. But even then, there's still that risk of human error that can be introduced, and that can happen either by not measuring something correctly, or it can be introduced by a contaminant that gets added into the batch every time that she goes to extract samples for testing. But by automating this process with the Ismatec Reglo ICC, then Tanya can potentially double her processing rate, or even more so, because she's leveraging the pump to do that addition of components to the media mixture and allowing the pump to draw the regular samples, and that allows her to focus on, and it frees her time to really focus on that sample analysis and to make those adjustments and improvements to the overall process, because some of those manual steps are being automated along the way. So that really is just a brief look at one of the hurdles that you might have to overcome at the research and development stage. So as we take a look again now at the BioPharma workflow and understand these different stages of development, once we move beyond the research and development stage, before we can get to the full out production, we've got the pilot plant stage. And in the pilot plant stage, the primary goal is really determining what that theoretical yield will be in the full production mode. And then we need to make sure that we're making those necessary process improvement steps along the way to get us as close as possible to what that theoretical yield could be. And those process improvement steps can be fine-tuning the growth media and that environmental recipe that we're employing. Anytime that you've gone from a small scale, even in your kitchen, perhaps when you're making something, we go from making a small scale recipe up to a larger scale. There are interesting things that can happen along the way that didn't happen at the small scale that begin to show up as they're enhanced by the larger collection of ingredients and mixtures and just the dynamics and physics of what has to happen at that point. So it's important that you take the time as you scale things up to both identify, observe and identify, and then mediate those issues that you might come across. And then also look for those potential roadblocks and the challenges that the production team at the next stage might experience when they migrate up to that full out production mode. So that big challenge is that time-consuming manual input and control. Remember that goal in mind is preparing for that full production process. And because you're making those fine-tuning stages that are during this pilot plant stage, one of the biggest challenges is all that manual intervention that's needed in order to properly monitor the media consumption, fluctuations of pH and temperature, and looking at the overall cellular health, and all of these things that you've worked so hard at the R&D stage to develop and get that initial process going. Now we're really, as we're scaling up, we're really having to monitor quite a bit at the same time to make sure that we haven't missed something along the way. And that big question that's in people's minds often at this stage is, really truly, how can I safely monitor all of these critical parameters? How can I look at everything that I have to monitor and then respond nimbly to ensure that the pilot batch cycle, that whole thing in the pilot plant that I'm trying to produce, that it continues to completion. I want it to go all the way to the end without having any interruption in the process or any loss of components. That's really what my goal is. So how do I safely monitor, and how do I do so without introducing contaminants? And to help with some of those challenges and help with that stage and all of the things that we have to do, then another tool that might possibly be considered is the MASTERFLEX LS process drive that features MASTERFLEX Live technology, and it's coupled with a Cytoflow pump head. And in order to walk us through how this tool could help us and really have a direct positive impact on users at that pilot plant stage, I'm going to turn things over to my colleague, Phil Nyring, and he's been one of the champions of this technology from its inception. So Phil, could you talk us through this a little bit?
I can, John. Thank you very much, and hopefully my audio issues have cleared up and everyone can hear me fine right now. But yeah, as John said, I'd like to switch from the hardware of pumping and fluid handling to perhaps some options for accessing and controlling that hardware. And that's really what MASTERFLEX Live is all about. MASTERFLEX Live, in its essence, it's a cloud-based, a cloud-based application for controlling and monitoring pumps across the LS and IP pump ranges. You saw one of our LS process drives depicted in the earlier slide, and on this particular slide, you see one of our LS standard digital drives. So it also has options in the IP range, with the IP range being a larger format pump with higher flow rate capabilities. But a little bit more about Live. Live is an accessible via a browser or mobile app, and it supports both iOS and Android devices. So the question is, what can MASTERFLEX Live do for you? And this slide gives you a little bit of a hint, in as much as MASTERFLEX Live offers you user-selectable notifications of the pump operational status. And those notifications are available through either text, email, or push notifications, and those are kind of standard in similar type formats. And that again is user selectable. You can select all or one or none in terms of the notifications. But that capability will notify you of the pump starting and stopping, for example, perhaps a power failure or other alarm condition, or when a programmed run is complete. So routine and non-routine aspects of the pump operation, you will be notified instantaneously wherever you happen to be relative to that pump. So in addition to the assurance of the pump operational awareness, MASTERFLEX Live enables what I call true remote control. So you can imagine being able to control and monitor your pump or pumps. MASTERFLEX Live can control any number of devices, whether it's on the bench in front of you or in a glove box or a hood, fume hood, a chamber, or even a clean room. And with MASTERFLEX Live, there's no need to physically enter the chamber or clean room for any routine changes to pump operation. And as John alluded to earlier, that minimizes the risk of you introducing contaminants into that chamber or clean room, but also, depending on the nature of the work you're doing, it can minimize the potential for any risk to personnel who might have to access those pumps if need be. And really, I hope that gives you a very, very high-level idea of what MASTERFLEX Live is capable of. There's a great deal more depth to it, but want to just kind of summarize all that by saying that MASTERFLEX Live is really a very powerful tool that you can apply in your applications, in your daily work. So the next thing I want to touch on, the next subject or product area I want to touch on is the MASTERFLEX LS and IP Cytoflow pumps, which again you saw depicted in that earlier slide. But the MASTERFLEX Cytoflow pumps are optimized to pump live cells, large molecules, and any other shear sensitive fluids, and fluid components or suspensions that are prone or could be prone to damage or alteration by the mechanics of pumping. That said, peristaltic pumps are inherently low shear. They are inherently capable of handling large molecules, but the LS and Cytoflow pumps have some design modifications that really optimize that level of functionality. And the assertions that I'm presenting you today, we have some third-party test data that we can provide upon request, but you see a little bit of the data provided on the slide in front of you, comparing the Cytoflow with the convex rollers, the green graph, with other types of peristaltic pumps, including what is typically present in a peristaltic pump, which is the flat occlusion or the fixed occlusion flat roller, the blue curve. So the Cytoflow heads, functionally, they rely on the interaction between a unique convex roller and a concave occlusion bed, which further minimizes the impact forces on the fluid as the pump operates. So they also incorporate large diameter rotors that enable you to deliver high flow rates at lower speeds. And lower speeds, or running the pump at the lowest possible speed to deliver your flow rate, will also optimize cell survivability, also optimize maintaining the integrity of the fluid that you happen to be pumping. So that's what I have on Cytoflow.
Thanks very much, Phil. I really appreciate you taking the time to walk us through that and help us understand some of those benefits that the MASTERFLEX Live, coupled with the Cytoflow technology, that those things can bring to the pilot plant environment and really help bring some opportunities for process improvements that people can experience. So now that we've briefly discussed those first two primary development stages, that initial discovery, research and development stage, and the pilot plant stage, I want us to take just a moment to talk through this last one, being really the one where the money is being made for most organizations, and that is that process and production side. Here in the process and production stage, the ultimate goal is to really establish a production level, repeatable process where you can regularly and repeatably produce that target product at a full production level, where you know everything that's happening, you understand all of the processes that are in place, and you know how to manage and mitigate the challenges. You know what those challenges are going to be, you know how to mitigate those, how to watch for them, and you have an expected process and an expected outcome. And for this stage, again, as with the previous two stages, automation is critical to achieving that process improvement that you're trying to do. Very critical to achieving those final results. And all of that is facilitated by the system components, those main system components that are employed throughout that production process of being fully networked together, and where there's a centralized control over all of the variety of pieces of equipment that are part of that process. And oftentimes that's driven by either Ethernet IP protocol or Profibus technologies that are network communication capabilities employed in bringing a bunch of equipment together in a centralized way. So, of course, one of the biggest challenges, as mentioned, is the biggest drivers that can fuel success at this stage of development is the full automation and control of all of the vital pieces of equipment. In the production setting, it's absolutely critical to keep things moving all the time while also monitoring things all the time. And to help us understand those challenges that can be faced at this stage of development when we're in the full production cycle, I want us to consider John Smith. So John, he represents a typical production engineer, and he is tasked with keeping that production process running smoothly and running predictably. And to do that, again, one of the things that's oftentimes employed is networking all the hardware together at a site level, so that one person or a team of people in a control center can keep an eye on all of the critical parameters. And typically that happens, again, using some network protocols that have been employed throughout the entire site. And so by fully automating the system, what that enables is a smaller team of individuals to nearly instantaneously respond to possible challenges that could happen. And also as a result, there's contamination that's minimized because through automation, you can avoid all of that manual intervention that would typically be needed without it, and allow sampling to happen, allow monitoring to take place, all without introducing contaminants into the workflow. And one of the tools that Cole-Parmer can bring to the table is the MASTERFLEX IP process pump. And this is a pump specifically that is coupled with advanced networking capabilities. And in order to help us better understand this particular tool and how it might be able to benefit you, I'm going to turn things over to my colleague, Mr. Colin Heathfield. Colin, would you help us?
Thank you, John. So, what does Mr. John Smith need to achieve? He's trying to increase flow rates to make it to a bigger scale. Scale up, very easy with a peristaltic pump. Sterility of chamber, very easy with a peristaltic pump. We can achieve with this IP range, we're looking at flow rates of 0.01 litres per minute, so the very low end, up to 19 litres per minute. The IP process drive will achieve what the process engineer needs for that production scale. It's got a 316 stainless steel casing with an aluminium anodized base. That means your casing is going to ensure that it fits in with your hygienic environment, with your washdown environment, and 316 stainless steel won't show corrosion or rusting. So this does tick the boxes in terms of fitting into your environment. But it's also got, coupled with, you have the controls, as you've had previously on the front, for the standard setting for dose, dispense, time, flow rates, etc. We now couple that with Ethernet IP or Profibus communication. We call these advanced communications. But what this means within the biotech industry is that it can communicate with a greater system and be communicated with by a greater system. It's got to be a two-way flow of information. So you still have the manual control on the keyboard at the front, but you can now integrate this unit into your automated system in your production environment and your hygienic stroke clean production environment. So the advanced communication does this by integrating itself with your system without additional firmware or hardware from the pump side. We also have another couple of options. If you look at this drawing, you see there's a small little wire sticking out to the pump head. It has what we call an open head sensor, so that if someone opens the pump head, it will tell the pump. The pump will tell your system via the advanced communication. This means, one, it stops the pump, so it's a health and safety supplement. And two, it means if your process is interfered with by someone accidentally opening the pump head, it will tell your system. This is all part and parcel of this two-way communication from the system to the pump and the pump to the system. It also has, or we can offer, a leak detection system, which means we can detect if there's fluid at the bottom of the pump head. It's additional to this, but that would then indicate if there's a tubing issue or a tubing problem. So suddenly the pump has a level of intelligence to work with your system that will actually ensure that the system will know what the pump is doing, not just tell it what to do. We recently had a situation with a customer where they wanted to set up a stainless steel pump and wanted to use this in a biotech application to work with their equipment. So they wanted to run a pump or several pumps to feed a reactor and to work with their equipment. The pump we used was these process pumps, and the reason they do so well, their equipment via Profibus feeding into the reactor, and I think something similar to a Siemens type control system, was giving outputs to control the pump to say, please do this at this rate, please do that at that rate. So controlling the feed to a very accurate rate, which is what we can do with the peristaltic pump. But the pump was also then going back and saying, I'm running, and I'm running at this rate. The pump was actually being controlled from their system, and their system was being controlled from supplementary equipment. So from flow meters and pH and other things within the system. So it becomes a very critical part of that intelligent system. And from this, it achieved the benefits that the customer needed, which is one, the MASTERFLEX tube. We've got 20 plus tubes we can put in these drives, and the tube is the pump. So this pump chamber, which is the tube, can be changed. Even we go up to gamma irradiation for sterility if required, coupled with non-contact monitoring and operation, and it was linked completely with their system. Not just their system telling the pump what to do, but the pump telling their system what it was doing and that it was running and that it was running as necessary. And this can be achieved, as I said, with the advanced communication, and we have the Ethernet IP or the Profibus as the versions to use on this. John.
Thanks very much, Colin. I appreciate it. Thanks for walking us through that and helping us understand this tool a little bit more in depth. Now, by the way, as we kind of wind down our presentation today, I just wanted to take a moment to remind you, please submit your questions. We've got a chat window that's there, and if you could submit them there, we are setting aside a little bit of time at the end of the presentation in order to answer some of the questions that have come in, but we recognize that there may very well be some questions that we can't get to. If there are some of those questions that are asked that we aren't able to address during the course of this presentation, as Joe mentioned at the beginning, we will be sure to summarize them together in an FAQ document. All of you who have registered have provided an email address, and we will make sure to email you that document so that you have a list of all the questions that were asked, as well as the responses to those questions that you can use for future reference. So thank you in advance for taking the time to provide those questions and giving us a chance to answer them. So as we wind things down, we would be remiss if we didn't discuss the impact on our working conditions that COVID-19 continues to have at a worldwide level. If you think about returning to work and all of this, and things have changed. Our normal, what used to be normal, has shifted to a new normal, and our challenges revolve around maintaining productivity whilst still adapting to this new normal that includes maintaining that safe social distancing from others in the process, and that That simply requires a change in how we do our work. It may impact the number of people that can be in a laboratory or on the production floor at any one given time. It might require people who were used to working during a normal workday shift to have to pick up a mid shift or a third shift and to keep things moving because of having to make adjustments and adaptations to maintaining social distancing and helping really to limit the amount of spread of COVID-19 that takes place in the workplace. So, and this is another instance where MasterFlex Live technology can truly be a benefit to you as we adapt, as all of us are adapting to this new normal. Because MasterFlex Live technology, as Phil mentioned, it allows for this complete, very secure control over pump operations from anywhere an internet connection is available. So that means that it's possible for workers, authorized workers, to continue working with their pump without physically being next to their pump. They don't have to be in that area where space may be constrained, where there may be a limit to the number of people that could be together, because you can physically control the pump from really anywhere. And so this helps those authorized users to maintain operation of their pump and maintain productivity, whether they're at their workstation or really from a home office. It doesn't really matter where you are. If you have an internet connection, then you can control that production. The operation of the pump. You can control what the pump is doing. You can monitor things that are happening with the pump and start and stop it and control the flow rate and do all of those things as you get inputs from the system. So that's just a side benefit to leveraging technology in our post-COVID environment. So as we've seen, there is some key takeaways. Really the big message is that there are ways that you can take some of today's most innovative pump technologies and really make them work for you. Whether it's in that Ismatec Reglo ICC to help you drive and automate that R&D process and be able to simultaneously add and tweak the mixture of the media that you're using in order to grow the cells while also monitoring for cellular health. There could be MasterFlex Live equipped drives that help you respond quickly to changing conditions that frequently happen in a pilot plant scenario, or even to facilitate working remotely like we just talked about. And then even with the advanced communication capabilities, that advanced networking capability of the Ethernet IP and Profibus enabled MasterFlex drives to support production automation and really help enhance what you're doing at the production side. Really, all in all, these are just tools that we're bringing to the table to help you see the possibility of experiencing some process improvements and also to help you mitigate risk along the way. And this are the things that some technology can help you achieve. And really, truly, all of us, the three of us, as well as our entire team of technical experts that are situated all around the globe, we would love to partner with you to help you determine what's the best mix of technology for you. What are the best tools that you might be able to utilize at your facility, in your organization, in your application that would ultimately benefit you the most. And as we mentioned at the beginning of the presentation, help you gain time back in your calendar and help you save money in the process. So with that in mind, we thought we'd take the remaining portion of our time together and leverage it to the next 10 minutes or so to maybe answer some questions that have come in. So Joe, could you help us with that?
Yeah, hey John. So we've got quite a lot of questions on the Reglo ICC. So I'm gonna read these out in the order that they appeared. Okay. So there's a question around can the Reglo ICC, can it be independent? Can it run independently on stop start timer and not just continuous?
Yes, it can. Yeah, you can absolutely run it, and there's software control that you can employ so that you can control each channel exactly what it's doing. So whether it's running in a start and stop mode for dispensing, so just a momentary dispense and then stopping, or if you want to run it continuously, you can do either one.
And then the question kind of led on to so on any of the 1 to 4 channels, can the flow rate be different in terms of seconds for every hour? So if you want to sort of answer the question in terms of, can each channel have its own flow rate?
Absolutely. Yeah. So really think of it like, let's say, for instance, you have a 4-channel Ismatec Rego ICC, then that 4-channel pump is actually like having 4 pumps in one. Each channel is being truly controlled independently. So if you want one channel to run at, say a feed rate of 2 milliliters per minute, and you want another one to trickle in something at 100 microliters a minute, and you want another one to go in the opposite direction and sample every 10 seconds a sample from your media mixture of 3 milliliters, and whatever it is that you want to achieve at a smaller scale. You have to keep in mind that the maximum flow rate that these can achieve is around 45 milliliters a minute or so. And so that means that you really are at a small benchtop laboratory scale. You're not at a process scale or a pilot plant scale. This really is a tool for that R&D small scale batch. But within that realm, within that scale, then yeah, every channel can truly operate like a fully digital, fully controllable pump, but do so in a very compact way. So that's a big benefit. It should note that this is patented proprietary technology. There's no other pump in the world that does what the Ismatec Rego ICC can do.
That's a really good answer. I'm just going to ask one more Reglo ICC question. I think that links to your answer perfectly well, but just in case. We had, can different samples be taken simultaneously on the Reglo ICC? And I think that's referring back to what you've explained, as in the channels working simultaneously.
Absolutely.
Yeah.
Yeah, for sure. In fact, if you wanted to, I mean, you could think of it really good to think outside the box. For instance, let's say at the R&D stage, let's say, for instance, you're wanting to, rather than monitoring one batch of production at a small scale, say you wanted to monitor 2 batches simultaneously. And so maybe you had a regular recipe, you've already figured out what the optimal media is or what that mix should be. And let's say you had a 4-channel Rego ICC, you could have it so that 2 of the channels were feeding and sampling from. So one channel feeding into chamber number 1, another channel sampling from chamber number 1, simultaneously another channel feeding into chamber number 2, and the 4th channel pulling a sample from chamber number 2. So you truly can really leverage this automation tool to really enhance the kind of testing that you can do during the research and development stage. Absolutely.
Okay, that's great, John. Thank you. There's a few MasterFlex Live questions on the board as well. Does the MasterFlex Live have the ability to show the activity of multiple pumps at the same time, like a workflow status chart?
Joe, I'll take that one. This is Phil. Hopefully you can hear me. My phone seems to be glitching on me, but yeah, it absolutely can. When you use MasterFlex Live, you establish a account that has secure password protected login. And within that account, you can add any number of pumps. And once those pumps are added to the account, you have full visibility via a dashboard as to what each one of those pumps is doing, and individual control panels that enable you to control or adjust the control parameters for the pumps individually. So hopefully that answers the question. There is some additional functionality in terms of being able to group and sync similar pumps, but yeah, hopefully that addresses the initial question.
Okay, that's great. I'm not sure if I'm going to get this right or not, but there's a question that says, will there be any IO or OQ available for the MasterFlex pump?
Yeah, I'll take that one too, Joe. That's fine. IQ, OQ documentation, we have some basic templates that we can provide to you that you can use to develop the IQ, OQ specifics for your equipment and your application and your facility. So depending on where you are in the world and who you would purchase MasterFlex pumps from, you can communicate through them, and we can help you out there.
Okay, that's great. And then there's a MasterFlex IP question around the the open head sensor. So does a head sensor stop when the pump head is open to stop the drive motor?
I can come in on that one. So when the pump head is open, so the pump head that we showed has 180-degree movement on the lever to open the pump head. But as soon as you move that lever part of the way, it stops the drive. So the pump head will stop the drive as soon as the pump head is opened. This then means that the rotating rotor within that pump head stops almost instantaneously. So there is no longer a moving part and a risk to people's fingers. This gives the health and safety side of things a big tick, because it stops the drive, it stops the movement, and it means it's safe within there. That then comes up on the pump display, pump stopped or drive stopped, and that will also, if used with the Ethernet IP or the Profibus communication, will send a signal to the system to say pump stopped. So when you close it again, depending on how you set it, will be whether it starts again or whether it waits for a confirmation command from the system or for you to push the key on the control panel. But it will, on opening the pump head, stop it.
Oh, that's great. That's great. Thank you. Thank you for that, Colin. So someone asked, can you explain or elaborate on why the tube is the pump?
It's one of my favorite sayings. When you're using a peristaltic pump, when you're using any pump, the pump chamber is the critical part. But with other technologies, the pump chamber is actually is there as a static, and there's something else working within it. Say a centrifuge, you've got a rotor, progressive cavity, you've got a worm drive, even a diaphragm, you've got the diaphragm moving. The tube is having to do the work. So to actually get the liquid into the pump and then just squeeze it in the tube and squeeze the tube pushes the pump out nice and easily. But the pump tube has to open up and pull this liquid in. So that pump tube is the critical part of the system, because it's the pump chamber, it's the component that's doing the work to pull the liquid in, and ultimately it's the component that's doing the work as it's squeezed to push the liquid out. So the reason I say the tube is the pump, because the tube is the critical part. And if you get the tube right in terms of material for chemical compatibility, size for the flow rate you're trying to achieve, and you combine that with the number of rotors on number of rollers on the rotor to give you the right number of pockets to give you accuracy for dispensing, then everything else will work. So as long as you get that tubing right, then your pump system will work. And then, as we said, and we've shown through this, from scalability, from your lab scale to your pilot plant to process, to scale up with a peristaltic pump is very easy, because you just use a bigger tube with a thicker wall. But these are tubes that we developed specifically for use within peristaltic pumps.
That's an amazing answer. Thank you.
Can I, Joe, if you don't mind, I wanted to just interject a quick question and ask Colin, maybe if you could use this just to help, and we can all chime in perhaps if we have any input, but one of the things that we highlighted at the beginning of the presentation was the whole idea of single-use capabilities, and perhaps how some of the technological tools that we were discussing might help facilitate things at a single-use side of things. So Colin, I'm going to ask if you could kind of start off that discussion a little bit and just highlight how a peristaltic pump is uniquely situated or uniquely positioned to really be being employed in a single-use type of environment.
That goes back, John, to the start of the conversation. The tube is the pump, so the tube is the pump chamber. The only thing the liquid is in contact with is the pump chamber, which is the inside of the tube. Because it's a tube, you do think of it as a consumable, as a wearing component, but also as a disposable component. So it means that if you need to change your pump chamber, You don't have to undo lots of bolts and pull things apart and clean a system down and sterilize everything. You literally open that pump head, take the tube out, put a new piece of tube in, and close the pump head again. That's literally putting a new pump into your system. Now, if you put that in terms of single use, so you need gamma irradiated sterility, which again is a tube we offer, you just change the tube each time and you've put a new pump tube. So if you're putting in a single use system with a mixing bag, with such, there is pressure sensors, and we do disposable pressure sensors that you can put in tube. We do disposable flow meters. The whole system to give feedback from a single use system is available, but the whole thing of single use is to do the one production run and change, and the peristaltic pump facilitates this because it's that changeable pump chamber, and it's the only pump technology that has a changeable pump chamber.
Thanks, Colin. Phil, do you have any input you wanted to add to that?
I do not. That was pretty comprehensive from Mr. Heathfield.
Yeah. I think the one thing I would
on pumps.
Yeah, and
Okay, well,
I would say one of just really quickly, one of the other side benefits, and this is just to help dovetail into what Colin mentioned, and that is simply because the tube is the pump, and because the tube is the only thing that touches the fluid. What that means by default is that the rest of the pump hardware doesn't touch the fluid. And so therefore, when you're thinking about that easy cleanability and easy transition, when you change the tube, you don't have to worry about cleaning the rollers and disassembling the pump head and all of that, because again, the only thing that the fluid is touching is the tubing. And I just wanted to help emphasize that. Joe, do you have another question you wanted to talk about?
Yeah, there's quite a few, and I was just thinking there's only 4 minutes, so I'm going to try and get a couple of these questions out to you. How many tubing formulations do you provide for the MASTERFLEX peristaltic pumps?
So I can take this one. We say 20 plus. With that, we cover most, I call it pump media, liquids, and then they cover a range of sizes. So we have chemical ones, we have bio ones, we have ones that last longer than others. So with each tube, when you come to pick the tube, discuss it with us. Let us bring our knowledge to the table and talk to you. Because there is so many tubes, there is almost nothing we can't do. I don't think I've actually come across in all my years of working for Cole Palmer a liquid that I couldn't pump with a peristaltic pump.
Yep.
We have special ones for oils. We have weldable tubes, so you can join and fix to others. And these are all tubes that I reiterate we have run and tested with our drives and pump heads. So these are all proven within our drives and pump heads. So it gives the added advantage of knowing that it's going to work as a pump in the pump tube, in the pump drive. That's the end of my answer.
Excellent. Thank you, Colin. This one's a kind of double-barreled question. What is the maximum pressure head that the IP process pump can provide? And why is it not possible to provide peak pressure head of an IP pump?
Sorry, Joe, can you just repeat that question? I missed a
I
couple
will.
words.
I will. I think the first half of the question is probably slightly more relevant. So what is the maximum pressure head that the IP process pump can provide?
Yeah, I mean, with the peristaltics in general, the sweet spot for pressure is between probably 0 and 40 psi, which would be up to, what is that, about between 3 and 4 bar.
2.5 bar.
Pressure.
Yeah,
Okay.
2.5 bars. So,
We do have some specialized pumps that can do up to 150 psi, which is roughly 10 bar, but there might be some flow range limitations on those. Hopefully that answers the question.
Okay, that's great. I mean, the second half, I'm not quite sure if I'm kind of conveying this correctly, but it just says why is it not possible to provide peak pressure head of IP pump? I'm not sure if that's if that's actually answerable or not. I'm not sure.
Yeah, I'm not quite sure I follow. So we might have to follow up on that one.
Yeah, sure. That's okay.
Joe,
Yes.
there was one point I didn't mention when I was answering about the different tube formulations. And that was, it's not just the tubing formulations, it's also the classifications we have on the tubings. We have tubings that are FDA approved, we have tubings that are USP class 6. So we meet regulations with the tubes as well. I just wanted to have an opportunity to mention that.
That's great. Thanks, Colin. That's really appreciated. Right, we've hit the end of the show, guys. Thank you so much for your insight and skill and the presentation itself. I just want to say to everyone attending, there is a very short survey that we'd very much appreciate you filling out, which should be visible on your screens now. As we said earlier, this is being recorded and a copy of this will be emailed out to each and every one of you. And following that, there will also be a documentation summarising all the questions that we received with the best answers that we can offer, and that will also be shared in due course. So I wish you all a good day, good afternoon, and a good morning to you all, and thank you very much for attending today. It's been really appreciated.
Yes, thank you.
Thank you very much.
Thank you very much, everybody.